All right, good morning, everyone. I changed my title slightly. In inland waters, it's mostly cyanobacteria that are producing our cyanotoxins or algal toxins. So today I'll be talking to you about cyanobacteria harmful algal blooms or cyanohabs. And so you probably recognize this image. This is the bloom that occurred a couple years ago in the western basin of Lake Erie. It resulted in 500,000 people in the Toledo area being without a source of drinking water on the shores of one of the largest lakes in the world. So we're starting to hear more stories along these lines, and there's building evidence that global change is starting to turn our lakes from kind of typical blue colors to these greenish hues, which are caused by these cyanobacteria harmful algal blooms, or cyanohabs. So much so that there was a special issue in the National Geographic entitled, "Are We Fertilizing Our World?" And this is an image from the Yellow Sea in China, which tends to give us our most extreme images of blooms. But we are seeing more and more occurrences of beach closures and issues associated with harmful algal blooms. We know that they have serious human health risks associated with them, which is often what grabs the media's attention. People and animals can be exposed to them, either through ingestion and inhalation during recreational activities, so swimming, fishing, those type of activities. We're starting to find more and more indications that they are getting into the fish. And so we're seeing some more publications coming out, and we're doing some work in Missouri related to this, to whether or not they're getting into fish tissues. And then when humans consume those fish tissues, whether or not those algal toxins are having human health impacts. We also know that in some cases they can aerosolize, and so inhalation of the air around these blooms is of concern. And of course, what most causes our attention, and the reason why they had to turn off the taps in Toledo, is consumption in drinking water. So cyanotoxins will be present in the cells themselves, the phytoplankton cells, and also dissolved in the water. And so most of the time what we do when our water is contaminated or we question the source is we boil it, right? That's what we do when we're traveling to countries and things like that. In this case, if you boil that water, then you're going to be releasing all the toxins that were contained in the cell. So it's actually going to increase the concentrations of toxins in the drinking water. So right now there's no really good massive way to remove algal toxins. Activated carbon techniques are successful, but not at the large scale such as Toledo was faced with. So we currently just have to turn off the taps when we see these high cyanotoxin concentrations coming in. A lot of the concern when we treat our waters, most of the time we're able to remove these things, but not in these extreme blooms. And so, again, media has been picking up on this. We're seeing cases happening in Florida this past summer, and we're hearing more and more places and times and extremes in these cyanohabs. And its particular concern here in the Midwest, this is an image showing cyanohabs in colors, and so obviously the hot red colors there are the most occurrences. And so we're seeing it happen predominantly in this region. And so in order to understand why we're seeing increases in this, we need to understand what was the mitigating factor. So how lakes got from these beautiful blue colors to these greenish hues that we're seeing a little more often. And so we can kind of check off things and go through the different factors that regulate phytoplankton growth. We know that they are photosynthesizers and primary producers, so they require sufficient quantities of light. So we often look at light conditions, and that's where conversations about turbidity and things like that come into play. One of the most seminal papers in cyanohab works is Blooms Like It Hot, by Huisman and Pearl, and so we know that they do better and compete against other phytoplankton in really warm temperatures, and we're seeing trends in that in terms of our surface water temperatures. And we also know that they're reliant upon nutrients, and we often think about the macronutrients, nitrogen and phosphorus in this case, but they do have a high dependence on iron and some other micronutrients as well. And then grazing, so that top-down effect. So whether or not it's zooplankton or as depicted here, invasive zebra mussels, these are having an impact on controlling those populations. It's often been associated with zebra mussels because, as it turns out, things that produce toxins taste bad, and so we tend to see them being rejected from inhalation of these zebra mussels, and so having zebra mussels present can sometimes increase the concentrations of these cyanohabs in our surface waters. And so there's this great diagram that Jim Elser came up with, and it's kind of a lineage to how we can kind of look at tackling why these things are happening, what's the pathway to promoting these blooms. So the first question is nutrient loading high? And looking again both at nitrogen and phosphorus. If it's yes, what's the ratio of that? Are the light conditions right? Again, they can't bloom if they don't have enough light, and so making sure all those things check out, and then whether or not they're being grazed upon by the higher food webs. And so if everything kind of checks out and it falls along that line, then it's pretty likely you're going to get these noxious cyanobacterial blooms. And obviously, this is a problem. Too much is obviously not appealing in terms of recreation, but it also is producing a ton of biomass, right? And so this has to go somewhere and decompose, and this is how we get to realms of Gulf hypoxia and dead zones in our lake systems. The other problem with cyanohabs is that they're the wrong type of phytoplankton for a healthy food web. They tend to be competitively advantageous in situations of low nitrogen conditions because they have the ability to produce these specialized cells shown on here as heterocysts, so they can fix atmospheric nitrogen. So in situations of low nitrogen conditions, they can literally grab it from the air, so they tend to dominate in those type of situations. They also have the ability to regulate their buoyancy, and so they can move up and down the water column, and so typically we see in lake systems nutrients being higher in the lower at the sediment-water interface, so they can go down and grab those nutrients, and then they can go back to the top and get the light that they need to be able to grow, and so they're winning in the competition with other types of phytoplankton for these attributes. And of course, then they also have the ability to produce toxins. The other implications of this is that they have a really low lipid content, which means that they're effectively like empty calories or junk food for higher trophic levels such as fisheries. And so in addition to being noxious in terms of quantity, they're also the bad type of phytoplankton to have in your water bodies. So as mentioned, it's a little more complicated than that, although only cyanobacteria have the ability to fix atmospheric nitrogen, not all of them do. And it's often this complexity between the species that happen to be blooming as to whether or not they're doing it because they have this advantage of nitrogen fixation or the toxins or something else that we haven't quantified yet. So for example, Anabina, which is one of our classic bloomers, which they've now changed the name on us, they like to do that, is a nitrogen fixer. So you can see that it has these heterocysts on it. It produces toxins as well. In contrast, Microcystis, which produces the toxin microcystin, which is what we hear about in Toledo and other places, cannot fix atmospheric nitrogen. But it does also produce toxins. And so it's the balance between these two properties that kind of keep us puzzling as to why these are occurring more often in more places. They're also not new. So cyanobacteria produce the oxygen you're breathing today. They're some of the oldest organisms on Earth, and they've always produced toxins. In fact, they were producing toxins before there was predators. So, theories on anti-predation, things like that, kind of fall apart when you go back historically. But in extreme cases, they can be more toxic than DDT. And so not only do they produce microcystin, which is probably what you've heard about most often in the news and media, they also produce other toxins like spermopsin, anatoxin, saxitoxins, etc. So different species produce different toxins, and some species produce multiple toxins. So there's a reason why we haven't got this whole thing figured out yet. Also, the toxins themselves are different in terms of their components and their impacts. The liver toxins or hepatotoxins that we hear about most often, like microcystin and spermopsin, are the ones that we hear about mostly associated with dog deaths and things like that. Exposure symptoms would be vomiting, diarrhea, fever, cramps. These is mostly recreational type things that we hear about in the media. There's also neurotoxins, and so anatoxins and saxitoxins can produce symptoms such as paralysis and seizures. And then there's the dermatoxins, so less deadly, but they can also be irritant to your skin and eyes and nose and other components. So I borrowed this slide from Jennifer Graham at USGS, who is the expert in cyanohabs, and it kind of just hopefully conveys how complicated this really is. So here's the four kind of top culprits in terms of cyanohabs, and there's images of them below, and then it associates them with each of the toxins that they produce. And so Anabina, for example, produces all four commonly measured toxins. So spermopsin, microcystin, anatoxins, and saxitoxins. also produces the dermatoxins, and then there's also this association with taste and odor compounds. So geosmin and MIB is kind of the earthy, musty flavor sometimes you can get from water, and those are also produced by these cyanohabs or cyanobacteria. So it's complicated. The taste and odor, although not toxic and leading to death, kind of are a gateway to being concerned about cyanotoxins. So geosmin itself tends to smell kind of grassy. If anyone's ever smelled grass cuttings around water bodies, it's probably algae that are producing this compound geosmin. There's also MIB, which is often commonly produced at the same time. And again, as with the toxins themselves, these are really difficult to remove with conventional technologies and often result in simply turning off the taps. And so when their water smells, they tend to be concerned. So it reduces public confidence in our drinking water supplies. And so a lot of the challenges we have related to cyanohabs is it's still a bit of a mystery, and it's aggravating that we've been studying them for this long and still can't tell anybody which water bodies are most at risk, whether or not if you see green water, if it's toxic, and if it is green and we think there's toxins, what toxins are there. There's a variety of them, and they don't correlate with each other, as I'll show you some data from Missouri that demonstrates that. We also don't know where they're coming from. As mentioned, they've always been there. It's not a new compound, but something is promoting their production in terms of the algal populations. And so looking at these factors that regulate their growth and or when and or they produce these toxins. We also don't know how long the toxins persist. And so these issues come up when we have situations like Toledo. How long do you keep the water turned off? When we start looking at biota and getting into fish tissues, right now in Missouri at least, they have a 7-day advisory limit, so you're not to consume fish from a water body that has appeared to have a bloom condition in it for 7 days. But is that based on evidence? Do we know that it's not going into those tissues beyond the 7 days? And often, as I said, what you perceive to be green water is not necessarily producing toxins, and vice versa. So it's not as simple as that. I'm part of a group, a global group, we're GLEON, a Global Lake Ecosystem Observatory Network, where we're trying to put together all the toxin monitoring data from across the globe to be able to definitively say that they're getting worse. Because we still don't have any kind of these long-term programs or any kind of general perspective. We perceive them to be getting worse, but we're hoping to be able to put some numbers behind that. So I'll talk to you a bit about the work that we've done on cyanohabs in the state of Missouri, so continuing on the Missouri theme. I started my position there as an assistant professor 2 years ago, and I was lucky enough to inherit both a government-funded and a citizen science program that have been ongoing for 25 to 30 years. So the first program, which is funded through the EPA 319 funds through our Missouri Department of Natural Resources, we call the Statewide Lake Assessment Program, or SLAP. So it's been going on since 1976, and in total we've collected about 9,000 surface samples at over 250 reservoirs in the state. This is accompanied, a partner project is our citizen science program, the Lakes of Missouri Volunteer Program, or LNVP. And this has been going on since 1992. We get citizens who live on water bodies and have access to boats to go out and collect water quality samples for us. They then bring them back to their kitchens. We provide them with some filtration apparatus. They preserve them in their freezers, and then we go and collect them and pick them up about twice a year and analyze them back in our lab at the University of Missouri. So this is, it's been widespread, and it has a longer seasonal cycle. So our summer program is based on undergraduate student labor, so that only runs from May to August, whereas the LNVP program, turns out retirees have a little more time than undergraduate students. And so they collect their samples anywhere from April right through to October. And so I'm going to be presenting some data that combines from these two data sets. And so this is a map of Missouri, and every little MU symbol you see there is a reservoir on which we have water quality data. So our cyanotoxin studies were really initiated actually by Jennifer Graham. She did her PhD with Jack Jones at the University of Missouri and collected microcystin data from 2004 to 2006. So that's kind of our historical comparison. In all cases, we use the ELISA method to be able to analyze the toxins. And then we've since picked up the monitoring again in 2015, and it's continuing to present. And it's expanding. So we originally were just looking at the toxin, the kind of low-hanging fruit toxin, microcystin, but in 2017 we expanded that to also look at splenospormopsin, and then anatoxin and saxitoxins were initiated this summer. As mentioned, we're also starting to look at toxins in fish tissues from an agricultural reservoir, and this is thanks to funding from our Water Resource Research Center through the USGS. We're able to start looking at this persistence and source question. We're also initiating a new citizen science program involving high school students called ROS, or Reservoir Observer Student Scientists, in which we're getting students across 5 states through the gradient in terms of ice cover to non-ice cover to collect year-round cyanotoxin samples for us. And so here's some of Jennifer's data from 2004 to 2006. So the map is the state of Missouri. I've down there showing the thresholds for finished drinking water. So this is after it has gone through the treatment process. And we're typically concerned if it gets around 1.5 and above micrograms per liter. And so you can see here there's this clear north to south gradient. And if anybody knows Missouri well, you'll know that our dominant land use in the north is agriculture. And in the south, it tends to be more forested Ozarks region. So we definitely see this correlation between, trophic status and land use being kind of more greenish and eutrophic in the north to our more clear waters happening down in the south. In 2016, we saw a similar story, so not much temporal change in that case, where again the highest thresholds, and some of these are exceeding these limits. It's also concerning because the northern part of the state, the groundwater is saline, and so they are forced to use these surface drinking water reservoirs as their primary source of water. And so the fact that these are happening all in the same place at the same time is a bit concerning. Our data from 2017 shows less of those high concentrations in the northern part of the state, but we did find it in 90% of the reservoirs that we sampled out of 107 reservoirs. And so this represented about half of the time sampled over this 4 to 6 times that we sampled during the summer months. When we look at this historically, my postdoc Ruchi Bhattacharya has been looking at this, and she compared when we saw these microcystin concentrations to the Palmer Drought Severity Index, so looking at drought conditions, and we definitely see that the significantly higher microcystin concentrations were happening during periods of drought, as impacted by that gray square. And so there seems to be some correlation with hydrology when it comes to these productions. So as I mentioned, the program had historically been microcystin concentrations, and then the USGS, Loftendahl in 2016 published this paper using the National Lake Assessment data, of which we participated in, and we were surprised to see that, that Missouri showed up as having high splenospormopsin concentrations. So that led to our doing the statewide monitoring program in 2017 as a result of seeing this. So this is our data from 2017 across the state, and as you can see, it's fairly low. It was only detected in 20% of our reservoirs. That's relative to the 90% we saw for microcystin. And it only happened 7% of the times that we sampled, again, 4 to 6 times during the summer. So we don't seem to see as high concentrations of splenospormopsin. We often think that green water or blooms are associated with the production of toxins, and so we wanted to kind of correlate both toxins, splenospormopsin on the one y-axis and microcystin on the other with the boxes, against chlorophyll A concentrations, which is our indicator for phytoplankton biomass. And so you can see here that we did have some exceedances. The dotted horizontal line is showing our threshold values, so in some cases we did see exceedances. But when we looked at the comparison between these, they both had a really weak relationship with chlorophyll A concentrations, which means that green water does not necessarily equate to toxic water. We definitely saw a more positive relationship, between microcystin. Interestingly enough, splenospormopsin showed a negative relationship with chlorophyll A concentrations, so meaning that the clearer waters had less splenospormopsin. When we look at it in terms of trophic status, and so clear lakes are oligotrophic, green lakes are eutrophic, and we look at them in relationship to chlorophyll to total phosphorus, we can see that most of our microcystin detects are happening up in that eutrophic range, whereas most of the splenospormopsin are happening again in those clear water systems. And so the next step was to start looking at light and seeing if that had an impact. And so here now the x-axis is the attenuation coefficient, or KD. But again, we didn't see very strong relationships between either toxin. And again, it was positive with microcystin and negative with splenospormopsin. And so we're going to continue. We're continuing to monitor for toxins now, actually, currently. But microcystin still seems to be the toxin of interest, at least in the state of Missouri. And it was positively correlated with high trophic status. So it tends to happen more often in the north where we have the agricultural land use. And so I'm afraid to tell you that my current conclusion is it's still a bit unpredictable, difficult to model, and the answer may be monitor everywhere all the time because you can't necessarily relate it to green water. And so hopefully we'll be able to soon present our results for anatoxin and saxitoxin, and then with the citizen science program, that year-round component, because we typically only monitor during the summer months, but we drink our water year-round. And so whether or not we're seeing toxins happening in those shoulder and winter seasons is of interest. And so I don't have any good indicators of cyanotoxins for you, but I'd like to acknowledge funding sources and welcome any questions.